Super. Thanks, Ingrid. That's great. Right, 1401. I think I'm going to give a brief. I'm going to give a very brief introduction and welcome, but I don't think anybody who arrives after now, I don't think they're going to miss anything too fundamental. So I'm just going to go ahead and start introducing. Oh, hey, Steve. Oh, Timmy. Ah, super. Oh, right. Sorry, I'm getting sidetracked by the attendees. Right. So, welcome everybody. Can you hear me? Okay, everybody. Yep, great. So, welcome to the next talk in our Micropaleontological Society speaker series. So, with many thanks to Ingrid and Diana and others who are organising. We've got these sessions booked in with speakers in our field in an attempt to bring people together and understand what's going on in our discipline around the world. And it's great to see so many people here. We had Carlos Jaramillo last time, who was speaking on the subject of digital palynology. And this slot, we've got Surangi Punyasena, who is going to speak on AI provides new insights into paleoecology and plant evolution. So we're sort of walking through a development in our field. So by way of introducing Surangi, so Dr. Punyasena is at the University of Illinois, Urbana-Champaign, and research in her lab studies the influence of climate on the composition, structure, and long-term evolution of lowland neotropical plant communities. Dr. Punyasena is a palynologist, proper. So research in her lab uses fossil pollen to document the response of plants to past climate variability. The key thing that Surangi is going to talk about today, and one of the key things that she's doing in her lab that's really important for us all to take notice of and think about, is that her research is seeking to reimagine the field of paleoecology and to expand the range of ecological and evolutionary hypotheses that can be addressed with data from pollen and spores, I should say. And she's seeking to reimagine that by thinking of the discipline of palynology as a source of big data. So that means increasing the throughput, the reproducibility, as well as the taxonomic resolution of the data that we gather as micropaleontologists. She's focused recently on developing microscopy and computational methods in our discipline. A lot of research has been done from her lab exploring different microscopy techniques, from regular transmitted light microscopy all the way through to electron microscopy and super resolution microscopy, as well as computational techniques to analyze those images. And the long-term goal of her lab's research is to transform the paleoecological analysis workflow right the way through from imaging all the way through to classification of pollen and spores into taxonomic groups and the interpretation of the data that are derived from those pollen and spores. So I'll repeat the title again, although you'll probably see it on the screen in a second. AI provides new insights into paleoecology and plant evolution. Surangi, thank you very much for agreeing to speak. We're all really looking forward to hearing what you have to say, and I'll hand over to you. At the end of this slot, there'll be hopefully some time for questions, and I'll look out in the chat as well as for hands up on Zoom in the normal fashion. Okay, thank you very much, Surangi. I'll go on mute now.
Okay. All right. All right. So hold on. Let me just get myself situated. All right. Hello, everyone. Hello? Okay, someone needs to mute, please.
And as a last plea to all attendees, can you make sure your microphones are muted, please? Thanks.
Thank you. So, hello, everyone. Glad that you're able to join us today. So, good morning, good afternoon, good evening, depending on where you're joining us from. So, today I will be talking about AI, or more specifically, deep learning and computer vision applications that we have developed here at the University of Illinois in collaboration with colleagues all over the world, some of whom who are able to join us here today, to better interpret the plant microfossil record. All right. So this talk is also about the journey we have taken to get to this point over the last decade and a half. And I'm using the word odyssey here very deliberately, not because I see my life as a Greek tragedy, but because when we began this journey, we did not have the tools that are available to us today. And it took a fair amount of exploration and development to reach the point we are now. So, and actually, it took us longer. I looked it up than Ulysses' original journey. So, to all the early career folks in the audience, I think the lesson to take away is really to try to embrace uncertainty and risk early in your career and enjoy the adventure as far as you can. Yep. So today, my talk, as Luke pointed out, will talk primarily on the fossil pollen record. So the regular attendees of this series will know that fossil pollen is an excellent source of information on the composition of ancient plant communities. And so it's used to date geologic sediments, reconstruct paleoclimates, and study plant ecology and evolution over thousands to many, many millions of years. Pollen counts are also used in forensics, right? The CSI applications of palynology, as well as monitor air quality and even in agricultural applications, particularly in sourcing honey. But as Luke mentioned, right, part of what we do in my lab is to also think of pollen as an ultimate source of big data. So we have 470 million years of plant history, billions upon billions of potential specimens preserved in the rock record. And there are a very long time series of pollen and spores all over the earth across a range of environment. And that allows us to really study plant communities, plant evolution, and climate in a way that very few other terrestrial records afford. So why isn't everyone a palynologist? And you probably all wake up asking yourself that same question. And part of it is because of some of the key obstacles in attaining palynological data. So first, right, is the issue of data throughput, and Carlos went over this in his talk last month, but primarily we have relatively low data acquisition rates, particularly if we compare ourselves to, for example, geochemists. It's a very highly specialized skill. So many of you who work with microfossils will know how many years it took you to get to the point where you're confident in your ability to consistently identify taxa. And essentially, our counting methods have remained unchanged for over a century. And as a result, we have relatively slow throughput in terms of data production. We also have issues in terms of data reproducibility. So we use primarily qualitative descriptions for standardizing our identifications. We don't really have formal mechanisms for reporting our accuracy, right, and our confidence in any given identification. And as a result, the uncertainty that occurs at the count level can't be propagated, right, in meta-analyses or models that use palynological data. So this is not to say that pollen IDs are bad, but because of these issues of reproducibility, we tend to be very conservative in our identifications because we're aiming for consistency above all else. And so that leads to the final limitation, which is taxonomic resolution. Because of this need for consistency and reproducibility, we end up basically identifying to the lowest common denominator. So species identification are often rare are absent. Sometimes we can identify species, very rarely. Some quaternary folks try. Often we're identifying at the genus, sometimes family, sometimes order level. So many deep time of morphotypes in particular, as a result are end up named but not necessarily assigned a biological affinity. And so as a result of this issue of taxonomy, we end up with very limited ecological and evolutionary interpretations. So we can't work with pollen data on the granular level that we may like. And so my lab for the last decade and a half or so has really focused on these three main hurdles in palynological research. And we're seeking to transform pollen data into a higher throughput big data discipline. We're trying to improve the consistency and accuracy of our identifications. And we're also trying to create methods for assigning biological names to morphological types and allow us to really interpret the fossil pollen and spore record in an ecological and evolutionary context in a way that we haven't really been able to before. And so the two methodological advances that I'm going to discuss today that address all three of these issues are the high throughput imaging. And Carlos talked a lot about this in his talk last time, including the work we've done in scanning entire slides to create virtual representations of palynological samples, as well as the development of many optical super resolution techniques. And then once we have these images, the floodgates open, and there are many different types of computer vision techniques that we can use to not only detect and segment images to identify where on a slide a pollen grain may occur, but also clustering and classification, which allows us to automate some of the IDs, right, and do it in a very quantitative and systematic way. And finally, we can use a lot of the features that can be extracted by deep learning models to essentially interpret the phylogenetic relationships, so evolutionary relationships, and the ecology of the ecology of a plant community. And so here, so in the talk today, I will start talking a little bit about some of the future work for phylogenetic and ecological inference that we see. Okay, and so like any true Odyssey, there is a large cast of heroes in this story. So I want to start with my acknowledgments. So colleagues at my microscopy core facilities at the University of Illinois played a large role in introducing me and my lab to the instruments that are now really central to our research. I also benefited from very long-term collaborations from the U.S. National Science Foundation's Innovations in Biological Imaging and Visualization Sandbox that put together mathematicians, computer scientists, and biologists together to really develop some of these key collaborations. And that's something that has benefited both me and my students and introduced us to many of the computational and statistical methods we use now. And I'm glad to see Shu here in the audience with us. And so my former students and postdocs, right, were also quite instrumental, including the Micropaleo Society's Luke Mander and Ingrid, who basically played a very large role in developing many of the imaging techniques that I will be discussing. And more recently, Mark Iliadame and Jennifer Fang developed really exquisitely multidisciplinary research projects, right, in close collaboration with Shu. And those will be the focus of my talk today. And for the future, we have an excellent group of collaborators, including Carlos, who we heard from last month, and Tima Donders, right? So glad to see Tima in the audience today too, that are really going to allow us to scale up the imaging and move this research forward. And with my colleagues at the National Center for Supercomputing Applications, or NCSA, here on the campus of the University of Illinois, we're developing the computational infrastructure we need to share our work with the wider community, and really, I think will hopefully benefit all of us in the long term. And I'm glad to see Sandeep in the audience today. So, okay, let's start with imaging. So slide scanning provides a digital archive of pollen samples that can be annotated and analyzed with computer vision methods. So here we have one of several slide scanning microscopes that I have available at Illinois and that we use to digitize slides in their entirety. And so here you can see the entire sample digitized at fairly high resolution, so up to 400 times magnification. The Hamamatsu Nanozoomer is the one we use frequently because of it's one of the faster scanning scopes for standard transmitted light microscopy. However, right, there is also a need to find, we also had a need to find more efficient ways to take higher resolution images, primarily because of higher resolution than transmitted light, that is. Simply because the morphological analyses that we can do are limited by the morphology we can see. So here is Luke's beautiful visualization of how low image resolution results in low taxonomic resolution, right? So we can only tell apart pollen grains by shape, for example. And as we increase our imaging resolution, we're actually able to increase our taxonomic resolution because we're able to see these nanoscale features that distinguish species from each other. And so Luke spent his youth at Illinois exploring some of the different ways that imaging affected the morphology we could see. And that included some really early explorations of optical um, um, micro, microscopy, super resolution microscopy. Sorry about these stray marks here, but this is the SR SIM, the super resolution scanning structured illumination microscope here. And you could see that we could see far more with a super resolution than we can understand for standard fluorescence. And in some ways comparable, right, to the texture that's evident under electron microscopy. And so let's see if let me see if I can clear those those highlighter marks. 1 second. No, I can't. Sorry. Sorry about that. Okay, pointer options. Here's pointer. All right. So the next step, right, was Ingrid taking this a lot further by looking at area scan optical super resolution. And she really perfected the imaging of this microscope method. And so this is another optical super resolution technique that allows us to image both the external and internal structure of pollen grains down to about 140 nanometers. And so doing so allowed us to compare modern and fossil pollen morphology in a way that is far more detailed and at much larger scale than had been possible before. And so Ingrid's dissertation showed that we could distinguish between morphotypes of the fossil legume Stridopolys, right? And this just really shows the importance of this approach because particularly when we leverage deep learning toolkits like convolutional neural networks or CNN, we're able to tell apart different morphotypes, again, at increased taxonomic resolution. So I'm just going to stop this for a second to see if I could clear this.
Yeah, so, sorry, if you go to there's a little box at the bottom that says annotation
Yeah.
with a pencil.
Uh,
If you click that and then click the three dots for options. Oh no, sorry. If you click the recycle bin,
okay.
I think have I done it?
Yeah,
Yeah,
I
I
think you
think
have.
cool.
Perfect.
Thanks.
Not sure
Okay.
how I did that, but anyway. All right, thank you so much.
Yep.
All right. Okay. So, all right. So then moving on, right? Once we have these images, what can we do? So one of the results of having these full scans of, of of palynological samples is that we're essentially allowed to we we can capture the entire image in its entirety, but we don't necessarily need all of the information in this large image. We're particularly interested in the pollen material that's in it, for example. And so one way to facilitate the identification of pollen within this larger image is to use detection and segmentation techniques. And so this is an example of how we can leverage some of these deep learning techniques like convolutional neural networks and other machine learning architectures. And this is work that was started by Shukang and has been taken up by Sandeep. And two of my former students participated in this, and you'll be seeing material from Strive, from Enrique Moreno and Carlos Jaramillo, as well as some material from Tima Donders in Utrecht. And so some of our first approaches to pollen detection and classification basically used a fairly simple UNet where we trained on annotations, human provided annotations of pollen, not pollen, to find pollen within an image stack, right? So this is coming out of something like the nanozoomer, and then using these cropped images, images that were cropped by the machine to train an additional classifier to tell us what type of pollen we were looking at. And so this is looking at material from Barrow, Colorado Island, from the modern rain, pollen rain traps that you see here today. And so Jennifer Feng took this work a little further. She refined these detection algorithms. And so here we see quaternary samples from Ecuador, courtesy of Tima Donders. And what you see, and here the steps are essentially we're annotating, we're training, right? Essentially teaching the distance from the edge for an object right here. And then essentially then here you see the softmax probabilities once the model is trained and its ability to detect the edges and center of the object. And this would be the output result. Obviously, when you train and test, you would be using different images in the actual analysis, but for illustrative purposes, right, this is essentially the training and the testing. Once Jennifer had completed her master's thesis, though, we discovered that there were actually a lot more, there were more powerful foundation models that were available that we could use to actually produce very similar results. And so we started to experiment and as we finished up the paper with these large foundation models. And so in particular, Meta, so this is the parent company of Facebook, has a model called Segment Anything, right? And so SA, or also known as SAM. And so SAM, the newest iteration, SAM2, is a fairly powerful foundation model that can essentially, without any a priori information, detect and segment objects from a larger image. And so what we first did was use the segment anything mode where we identified the center of our objects of interest, which were produced here, right, with our pollen detection models. And then and used SAM to essentially segment or remove the background to clean up, right, samples for later classification training models. However, Segment Anything also has an everything mode. And this is really when we realized that the game for automated palynology really has shifted. So rather than training these bespoke pollen detection models, which really rely on human annotations and are very sensitive to differences in image quality, image type, whether or not you've stained your pollen grains, for example, Segment Anything can basically work with any image pretty much off the shelf. And so here you see some of Carlos's images from the Trans-Amazon Drilling Project. And what we have are crops that are fed through the Segment Anything model. And so Sam is able to detect all these objects. So every different color is a different object, right? Including the background, right? Being a separate object. And so essentially an entire image can be deconstructed by these foundational models with no prior training on what is and isn't of interest. And so what we have been doing is experimenting with segment anything for the segmentation of these large pollen sample scans. And so these are the four basic steps within the analysis. We essentially crop our nanozoomer or NDPI file into overlapping tiles of all the Z planes. And so depending on how many axial planes, it can be anything from 9 to 25 planes. For 25 planes, it takes about 55 seconds to crop all of the Z planes in squares of about 2048 by 2048 pixels. Okay, once we have these crops, we essentially have the square crops that we can feed into our analysis. We compress all that information using maximum intensity projection into our entire 25 plane stack becomes a single image, and that is fed into SAM2 using the everything mode. And you can see that most of the time is in the cropping, and very little time is really needed for the segmentation of these individual tiles. Once we have these segmented tiles, we save these segments, all the segment images for downstream analyses that include clustering and future classification. And the saving also takes more time than the actual segmentation itself. And so this is an example, and this is a view from the NanoZoomer soft NDPI view software. And so here you see the human annotations that Carlos kindly shared. And so in purple, and you could see here about 4 tiles worth of annotations from our SAM segmentation model. And what the first thing you'll notice is that I haven't displayed all of the red dots simply because it's very hard to for the software to process these many annotations and also for us to visualize it. But you can see that basically every object in this case that is approximately pollen sized is being segmented as opposed to just pollen. And I'll show you that in a second. So the density of segmentations is obviously higher, but we're casting a wide net, right? First, we want to start by identifying all the objects in an image, then we decide whether or not we're interested in it. And so what we found when we did this analysis, and we used something called intersection over union to identify where things were the same or shared with the human annotations, that anything with overlap of about 50%. And you'll see why that overlap is a little bit low, right, in a minute, because of the humans, not because of the machine. We were essentially detecting every object of interest identified by the human expert. And so this is an example zoomed in of what of these annotations. And so in red, you see everything that was detected as an object of interest by SAM. And here you see pollen green, and here you see the human annotation. And you could see that the human annotations were largely larger, right? Not as tightly bound. And this is why there was less overlap than you would expect with the annotations, but I would call this an exact match. Similarly, right here we have 2 human expert annotations, and you see the bounding box that SAM provides, right? And if we look closer at that, you could see that human annotations are always much larger, so not quite as tightly bound. And then you could see here that in this case, the human annotation also included something that wasn't of interest, right? So unidentifiable palynological material, but SAM is able to detect, right, and distinguish between the two very easily. Similarly, right, another view, I wanted to zoom in on this identification here. You can see here that the human annotation included both pollen and non-pollen material, and that it's overlapping, right, which made it difficult for the human annotator, but segment anything can distinguish between the two, right? And both of these are essentially segmented out as separate objects. Okay, so once we have all of these, right, what do we do? And so this is work that's in progress. And so I don't have our most recent results to show you, but I will show you what Sandeep has been doing, some of the work that we presented about a year ago on this process. And so this is, and so essentially what we're doing is going from these unsupervised detections, so using a foundation model, to also doing these unsupervised clusters. So again, without any training, asking our deep learning models to identify what looks alike and create clusters for us that we could potentially use later for annotations, right? It's much easier to label a group of images than it is to label images one by one, and that will allow us to refine our training models in the future. And so the models that we use for this particular result, the particular analysis that I'm going to show you is something called a swim transformer. I'm not going to get into the details of the architecture, but really the model, the architecture doesn't really matter. It's the pipeline and the logic that's actually really what's important. And so we're experimenting with several different architectures to see which give us the best unsupervised clusters. And this is work that Shu and his lab is also is essentially focusing on. And so the examples that I'll show you in a minute come from 6 Quaternion Ecuador slides, courtesy of Tim Hernandez. So we were able to detect every object on it. So these are not pollen detections, just general detections. So over 36,000 detections across these 6 slides. And we subsampled 1,000 of those. Sandeep essentially picked the most in-focused image of this image stack, forward passed it through this pre-trained, right, swim transformer. Again, this is the architecture doesn't matter in this particular case. And then we use PCA to reduce the dimensionality of this many thousand column matrix, right, and pick the 163 principal components that describe most of the morphological variants, and then ran a clustering algorithm, in this case k-means, to essentially identify the major clusters in the analysis. And so these are the 16 major clusters that were identified off of these 6 particular slides. And what you can see, right, in this analysis is that of the objects that the model sees a lot of, it becomes really good at creating clusters. So this is Lycopodium, right, added as a marker spore. Lots of Lycopodium in quaternary samples. As a result, the machine is very, very good at identifying different Lycopodium types. So very clean clusters of something like Lycopodium. Also in these samples, a lot of allness. Not everything here is allness, but the majority are. And so what you see here, right, is that objects that the computer can see has seen a lot of it can cluster without any a priori knowledge, right, that this is allness, which is very, very cool. And so for annotation purposes, you can go in later and say, look, right, this is allness. And then next, right, is a cluster where the clustering actually failed. So superficially, these look very similar, but what you see is we have some things like Cyberaceae, we have maybe Moraceae in here, we have grasses in here. And so these are all things that have relatively thin excites or are lighter appearing in color, but not morphologically similar at all. And the reason this fit analysis failed is because it was size agnostic, right? We didn't introduce relative size into the analysis. And so our current work is trying to improve upon these clustering results by reintroducing size into the analysis and keeping that a standard for the clustering. And that hopefully will improve our clusters and make them a little cleaner. Okay. Next, I will try to quickly go into what we've been doing with phylogenetic inference. And so this is really just shows the power of machine learning and deep learning beyond just simple classification tasks. And this is work that was done by Mark-Elia Dami, who should be here in the audience today too, working closely with Xu Kang. And I'll also show some samples from Carolyn Stromberg's labs as well, showing that this technique does not only just apply to pollen, but can be used also with other microfossils like phytoliths. And so this is the structure of a convolutional neural network, right? So essentially what you have is an image that's input into the model, convolutional layers that summarize, right? And pooling layers that essentially summarize the convolutional layer, this iterative convolution pooling, depending on how many layers you go, right? It could be anywhere from 34 for resonant 34 to 101 for a resnext 101. But essentially, you have these pooling and convolutional layers that leads to a fully connected layer that is a link to actual labels, right? And that produces the output or classifications, for example, pollen, not pollen, or taxonomic IDs or what have you. But the thing to note, right, is although we treat convolutional neural networks as black boxes, in the end, they're just quantitative summaries of an image. So the penultimate layer, so the next to last layer of a neural network, a CNN, for example, will essentially provide a numerical vector that describes the image in its entirety. And so this is what we call a feature vector. And feature vectors are really, really powerful in interpreting morphology, quantifying morphology, and understanding morphological diversity. And so Mark really showed what we can do with phylogenetic inference in this respect. So this is the analysis that he published as part of his doctoral research. And so what you see here, right, is that we have three neural networks, three convolutional neural networks, one that uses the entire image, in this case of Podocarpus, one that essentially looks at cross-sectional images, right? We can get internal structure from our super resolution images. And then the final image, which is looking at patches, so portions of the image. So we're looking at the surface and shape, we're looking at the internal structure, and we're looking at textural differences. Each of these CNNs produces a feature vector that essentially can be added to a fuse model, right, for classification. And so we can just use a novelty detector, and I won't get into this to decide whether it's something we've seen before or not. If it's something we've already seen before, no, it's not novel. We can do a standard K-way classification. However, in the case of most fossils, right, it's going to be something that we haven't seen before. We haven't trained our models before. So what do we do then? Again, we can take this penultimate layer, these feature vectors that are produced by neural networks, create a concatenated vector that essentially describes the morphology, external, internal texture, and train a second model that essentially reinterprets these morphological features in this case, a phylogenetic context. And this is what we call a phylogenetic embedding model. And so what our model did was take, right, this is a TSNA visualization of two dimensions of the feature vectors, right? And so before training on phylogenetic distance and after training on phylogenetic distance. And so what you can see is that before training on phylogenetic distance, here there was very little there was some structure, but not very much phylogenetic structure in the morphological features that were being identified by our deep learning models. But following, right, training on phylogenetic distance, the embedding model is able to produce features that essentially capture some of the phylogenetic variants in the morphology of our samples. And so here you could see, right, the two subfamilies of, subgenera of Podocarpus being identified here. So And so once we have that, those feature vectors then, and this was really a beautiful insight by Mark, can be used to produce a phylogenetic tree using Bayesian inference. And so here is the result of that. And so this is a phylogenetic tree that was essentially trained or produced solely with morphological information. So we're taking modern pollen samples, we're interpreting the morphology in a phylogenetic context, and then we can use those features that are identified by this model trained in a phylogenetic context to reconstruct the molecular tree that essentially informed, right, our ground truth of the evolutionary relationships in its entirety. So our relationships are basically the same as the molecular models that we use, or the molecular, sorry, phylogenies that we use, but the benefit of including morphology is essentially that we can add this to, we can add our fossils within, right, using where we don't have any genetic information, right, into the same phylogeny. And so what you can see here is we get the results we would expect, where the younger Panamanian fossils are related to modern neotropical species, and where our older Peruvian fossils and our oldest Colombian fossil, right, are essentially outgroups to the subgenus that includes the um, neotropical species of our analysis. And where the uncertainty is greatest is with Podocarpus salingus, whose placement actually is not known very well. And also an uncertain placement is our Panamanian fossil, which was so deep, badly corroded, it was the example that sort of proved the rule, where we had very little morphological placement to place it with any certainty. So this was really a really powerful demonstration of what we can do with deep learning and morphology, and our ability to really interpret this evolutionary placement of what we would have called podocarpides, right? And lumped together under a single umbrella. And then to show, and then just to show what we can, that this does not apply just to phytoliths, um, where this is an example, right? Um, um, from Ben Lloyd's work, where we can essentially use very similar technique to interpret the placement of phytoliths, matching, right, depending on the level of uncertainty, the exact placement of the most recent common ancestor, and also being able to differentiate between uh, morphologies that have a lot of phylogenetic information, the polylobate form, and forms that have very little phylogenetic information, which is this more plesiomorphic cross form. And then when we apply this to fossils, right, there is a potential to potentially get slightly better taxonomic resolution than our manual IDs. And so this is work that's ongoing, and Ben was able to present that at GSA earlier this month, if you were able to catch that. And then I'm just going to conclude with this look of, or expanding, right, the kinds of questions we can ask with pollen using these deep learning characteristics. And again, this is work that Mark did as part of his PhD. And it's really an elegant look at how we can interpret morphology in this case, an ecological versus an evolutionary framework. And to this, right, we go to grasses. And so one of the reasons why phytoliths are used in paleoecological analyses is because grass pollen looks like this, meaning that all 13,000 or so species, the pollen looks very, very similar with very small differences in pore structure and exine texture. But what Mark was able to show is that if we use these super resolution images plus neural networks, we can actually interpret some of the morphology at a much finer scale. And so he trained a model. So in this case, two models, one looking at just the whole image, one looking at the patches, right? So we're not looking at the internal structure so much here. So this is 60 modern species that represent a fair number of the um, taxa of Mount Kenya, which is a lake that we were interested in interpreting the palynological record of. And he developed a semi-supervised learning approach that include fossil specimens using a 101 layer neural network. And so essentially, he was able to use the features that were produced by these neural networks to reconstruct diversity, morphological diversity, and show that it approximated um, morphological diversity approximated palynological or taxonomic diversity using the entropy, right, the distribution of the morphological features. And so he was able to show that these features did actually capture taxonomic diversity using simulated communities. So if you divide your data set in half, train on 30 of these species, introduce the other 30 species as new random assemblies, we're able to produce a distribution, right, of morphological features and calculate the entropy from that. And so the entropy, the kernel density distribution, right, here that you see was essentially correlated. Um, the entropy of that distribution is correlated to Shannon diversity or Shannon entropy. So it was a very effective metric, right, of taxonomic diversity. And Mark demonstrated this further because we had five surface samples from five lakes on an elevational gradient for which we had some information about what the grass diversity was. This is on the slopes of Mount Kenya. And so what we could, and what you can see is depending on the smoothing, right, you can essentially capture that same gradient using morphological features of the subfossil samples, which is pretty cool. So we can use morphology to estimate taxonomic diversity. Mark went one step further to see whether or not he could tell the difference between C3 and C4 grasses. And this is the result, right? Where essentially he was able to use, again, reduced dimensionality of CNN learned features, train a classifier to tell apart C3 versus C4 pollen grades. And so one thing that Mark noticed right away is that if you look at the morphospace distribution of C3 and C4 grasses um, generated, right, as a result of these CNN features, they're quite distinct. He also noticed that the pixel intensities tended to be higher for C3 grasses, and also the entropy of the surface texture, right? So the morphological entropy of the texture itself was also more complex within C3 grasses. So there is actually a morphological difference in C3 versus C4 grasses, very subtle, that is related to something about differences in the X sign, either its thickness, its texture, its complexity. And we showed that actually that this was very similar to work that other folks had picked up on and maybe not interpreted in quite the same way. So this is work by Kaksha Wei, where she really did very detailed morphological measurements of different grasses. And so this is looking at a completely different taxonomic assemblage, right, of grasses. But again, she saw the same thing where the exine thickness of C3 grasses tends to be larger than that of C4 grasses. So there is some morphological differences within the pollen that allows us to tell the difference between C3 and C4. And so what we have, when we apply, right, these models to a long-term sediment record from Mount Kenya, we're able then to reconstruct some of the diversity dynamics in a way that we couldn't do before from grass pollen. And so what we see here, right, is our diversity estimates. And this is looking at just PC1 of the CNN features, PC1 1 and 2, PC1 and 2 and 3. And PC1 is highly correlated with CO2 and temperature, which is very interesting to see. So grass diversity increasing as temperatures rise, CO2 increases. The next thing we saw is that when Mark reconstructed the fraction of C4 grasses, that the trend was messy, but was positively correlated with other paleo records from completely different sources. So estimates from Ficken et al. Using grass cuticle and Urban et al. 2015 looking at isotope data. And so, so this is single pollen isotope data, which is fairly noisy, but the same general trend, right, of this sort of decrease from the last glacial maximum is apparent, which was also very interesting to see. The morphospace that is covered by these grasses, I don't think I have enough time to go through, but essentially shifts through time. And we do see an assemblage that seems relatively unique to the last glacial maximum. And so I just wanted to end, I'm sorry, I've gone a little bit long, on the work that we're going to do on developing community platforms, because we really want to make these tools available for all palynological research. And so this is work that Sandeep is heading, right, in collaboration with the National Center for Supercomputing Applications, as well as our long-term collaborators, Shukang Trellis Fox and Carlos Hadamio. And so what we're going to do is we're going to leverage the supercomputing power of the National Center for Supercomputing Applications and the software tools that they've developed for creating customizable data ingestion and analysis frameworks. And so Clouder is the platform that we're going to use. Its icon is this cute little cat, but basically you can think of it as a sort of a Dropbox, right, for data where you're able to upload the data, which triggers certain analysis pipelines, which essentially automates a lot of the work that I showed you today. So including potentially segmentation of entire slide scans, clustering, and those types of analyses. And so we're still in the early stages. We just got funded this fall, and we are going to move forward, I think, over the next 2 years to really make this a reality. So I hope I'll be able to come back in a couple of years and let you know how this is going and how you can use these tools in your research as well. And so with that, right, pollen microfossils are everywhere. They're abundant but underutilized. Computer vision really can solve some of the key problems in taxonomic resolution, reproducibility, and throughput. Machine learned features can be used in really new and creative ways. So we can work with morphology in a way we haven't before, which leads to my main conclusion that machine learning is really making morphology cool again in a way that we really did not anticipate. And so really the time to act is now, because there are these zero-shot segmentation models, right? These foundational models that essentially don't require any a priori training. These are models that we can leverage for our analyses. With enough imaging, with enough investment in computing infrastructure, we can actually automate many of these steps that we do in microfossil analysis, and we can focus on the interesting part, which is the interpretation, right, and the analysis. And we can essentially, we need to do this, we need to share resources. We need to share images of our samples. We need to share the metadata. We need to share these computing platforms. And I hope, right, that everyone here in the audience will join in this work and try to create a really community-minded approach, right, to palynological analysis. With that, there are many, many people that I'd like to thank, many of whom were mentioned in this talk. And if anyone is interested, we do have postdoctoral positions available in this research, including development of these analysis pipelines, as well as a project with Luke looking at early angiosperm evolution, if evolution is more your thing. So with that, I guess I left a couple of minutes for questions. So thank you very much.
Applause. Many thanks, Surangi.
So
Um,
sorry about that. Like, I can't believe I messed up the slides, but that's okay.
it's all great. Many thanks.
Yeah.
I think will open the floor immediately for questions. Thanks for finishing with time for questions. It's really useful. Oh yeah, somebody's commented in the chat. Yeah, machine learning is making morphology cool again. Is it possible to apply the same idea to faunal fossils like forams?
Yes, right. So anything where we can effectively capture the morphology is fair game, right? Right now, it's very hard to work with three-dimensional images, which is why, if you notice, we're using a lot of 2D representations of 3D data. And I think once 3D convolutional neural networks, 3D models of image for computer vision come on to the fore, that's really going to change things, the world for palynology forums where three-dimensional information is really important. But my understanding of forams is that there are canonical orientations that are imaged in 2D. And if that is enough for an expert to identify it, then that is enough to train a model on.
Mm. Yeah. Okay. Thank you. Yeah. But I think that notion of standardizing orientation is an important one for that group of organisms. Another question in the chat: how will you count in fossil samples for modern contamination and reworked material? Yeah. So I guess the question is, yeah, how do you account for modern contamination and reworked material in fossil
Well, I don't
images?
think it would be that different from how we do it now, right? So part of how we identify contamination, either from much younger or much older material, is usually based on changes in the sort of the exime, either its coloration, its morphology, either it looks too perfect or it's crumpled. So all these models do is cluster like, right? So they're basically doing the same thing we would do if I were to give you 100 images and say cluster everything that looks alike to you. What's really impressive is that these foundation models have reached a point where we don't have to tell it what is important to cluster like, where it intuitively knows what looks alike, similar to the way we do. Um, and so if you remember the example where the clustering failed, right, that's where we were not providing essential information, which was size. And
Mm,
so as a result, we were clustering things based more on color and texture, or which wasn't as informative as shape and size, right, in that analysis. So I think you would be able to basically identify clusters of reworked material. You would identify clusters of contamination if it looks different. But if it
mm.
didn't look different, then obviously that would be a problem with manual IDs as well.
There's a good question from Steve Stukins, I think, in the chat. What are your pinch points in the workflow that's currently creating that's prevent? What are the pinch points in the workflow that's currently preventing you from creating huge amounts of data? I
Yeah.
think is the question.
Yeah, that's a very, very good question. Part of was like, so this is not easy or cheap, right, at the moment, right? And this is the Greek tragedy aspect of this. um, that said, right, we're planning for a future where it will be easier and cheaper. And so I know that Steve had a question about like resource demands of these models. And I did want to say that training model foundation models themselves are quite expensive, but forward passing, right, using models that are already trained are relatively cheap. And I'm working with NCSA um, as we're using pollen as an example of something where all we need is the brute force throughput, right? We don't need fancy GPUs to do that kind of work. And so maybe that could be done more efficiently than we are now. And that's a cyber infrastructure thing that I'll let the computer scientists worry about. Um,
Yeah,
so,
there's a
but they, yeah,
yeah
but the other
there's a
pinch point,
related question from Carlos in the chat
yeah.
that's once you SAM to segment the a scan slide, what's the size of the image? Can you run it all this on your laptop?
Yes, actually. So, okay. So, yeah, the pinch points are really the segmentation portion takes a lot simply because of it sounds ridiculous, but the actual deep learning models are quite, the foundational model, SAM, is very, very quick. But the opening the image, creating the tiles, saving that material is what actually takes a lot of time. And so a lot of that open microscopy software only works on CPUs, which is far less efficient and as fast as GPUs. So if there were microscopy companies were to provide APIs which made it available for us to open their images, their proprietary formats in a very easy way, that would actually improve it because the NDPI images are wonderful in how efficient they are in terms of their size for the amount of detail, I would say, right? If you turned it into a TIFF image, it would explode the size. But it's harder to manipulate than these standard formats like PNG or TIFF. So that's one of the pinch points, right? Basically,
Mm,
going from the microscopy image into a tile that can be interpreted. And so that's computationally quite expensive.
mm.
Um, and then the analysis itself is because it's run on GPUs can be relatively efficient, and the clustering itself could also potentially be efficient. The question now is deciding right what is the but I think we're not at the point we can run it on a desktop simply because we'll have so many segmented objects. So if you had an easy way to filter out everything to just pollen, then maybe you can just do the analysis so on. But I think the problem is really dealing with, sorting through, we're casting a wide net and sorting through everything. And so I think until we improve that filtering process, then it'll probably be a little too much to cluster. If we tried to cluster all 36,000 objects, it would be
It's
really hard.
substantial.
So yeah. So yeah.
Okay,
So
it's trickled past three now. Sarangi, thank you very much again for speaking to us. Really, really appreciate your time. Thank you for a really, really stimulating and interesting talk. Really great. So thanks again. Yeah, some applause, definitely lots of applause on the screen.
thank you.
Um, our next talk will be on November the 24th by Evelyn Kustacher, who's going to speak on the evolution of the Kungurian flora in the Paleotropics. Okay, many, many thanks. I will close the meeting there. I think it's been recorded. I don't know where the link will be. I think it will be on our page, but I think an email will go round.